它的题目是Hemostereogenic enzyme genes in brain of protogenous grappers, possible role in sex differentiation. 欢迎他。
Good evening to everybody. I would like to present the entitled Neurosteroidogenic enzyme gene in brain of protogenous grouper, Epinephrus coitus. We compare this expression of gene in possible role in sex differentiation of grouper. So this is the content of the talk. So what is sex differentiation? The sex differentiation is the development of gonad and the differentiation into an ovary or testis from undifferentiated gonad to the testis or ovary is called sex differentiation. This is normally occurred in the mammals. It's also called gonadogenesis. So what about the teleus? The teleus having variety of the reproductive strategies that is called hermaphroditism, which means the fish can sex change from male to female or female to male. The hermaphroditism is divided into two parts: the sequential hermaphrodites and simultaneous hermaphrodites. The sequential hermaphrodites is a protokinase. This is the grouper fish, which sex change from female to male, and protandrous sex change from male to female. Here is some example of the hermaphrodites: grouper fish and black porky and butter hamlets. So the development of ontogeny of sexual dimorphism in the brain has been influenced two factors. One is a genetic influence and another one is the hormonal influence. By the genetic influence, the XX or XY chromosomes are playing an important role in the brain to differentiate the brain. But come to the hormonal influence. The hormones influence to the brain for cell migration, cell differentiation, cell death, axon guidance, and synaptogenesis. And the important physiological function is the neurogenesis. We are focusing on the neurogenesis in the brain of grouper. So this is the biosynthetic pathway of steroidogenic enzyme genes in the brain. Here you can see the cholesterol has been converted from outer to inner mitochondrial member by the star protein. And after the cholesterol has been converted to serial enzyme, CYP17 and 3-beta-HSD and aromatase. And aromatase doing important role in the steroidogenic enzyme pathway. It's converting testosterone to estradiol, and estradiol having a main physiological function of the brain. So in the early 1970s, the hypothesis proposed that the testosterone converted to estradiol in the brain by the enzyme aromatase. After that, in the homeothermic vertebrates like birds and mammals, the aromatase activity is more abundant in the diencephalon and limbic areas. And more recently, in teleus, they confirmed that the aromatase activity is 100 to 1000 times higher than to the mammals. Also, very recently, the brain aromatase and aromatase activity abundant in the radial glial cell of the teleus. Here you see. So normally the aromatase has been influenced by the estradiol during early development due to the estrogen receptor present on the promoter region of the aromatase. There are three kinds of estrogen receptor identified from the human red and quail fish. It's called estrogen receptor alpha, beta, and beta 2. This is the nuclear receptor, estrogen receptor. Out of this, there are two kinds of membrane estrogen receptor. It's called GPR30 and ERX. It's expressed in the brain and uterus. This is the molecular functions of the estrogen receptor and the brain. So the hypothesis, two questions in the present study. The first one, the de novo synthesis of estradiol have an impact on the brain development during gonadal sex differentiation. And the second question, the peak expression of this steroidogenic enzyme genes between female and male fish, it has a similar expression or not? So this is our experimental fish grouper. It's a hermaphroditism, conversion from female to male, and protokinase and monandrate. There are a lot of uses in the human. It's using in the fisheries, commercial, and it's very useful for the mariculture. This is the distribution of grouper, and mostly in Asian countries like Taiwan, Vietnam, Indonesia, and some part of in Australia. So we collected the sample from undifferentiated period to differentiated period, almost 6 points. We collect the brain part like forebrain, midbrain, hypothalamus, and hindbrain from each point. And this is the materials of our present study. So first part of the materials, we collect the sample for anatomical studies like immunohistochemistry and in situ hybridization. And another set of samples we're using for the molecular cloning. So this is the identity of clone sequence of the group of steroidogenic enzyme genes we compare with other TDS species. Here you can see all the steroidogenic enzyme genes have more than 85% with other TDS species, especially in the black coatie, it having more than 85%. When compared to the human species, it having more than 65% identity. So we did the developmental changes of the gonad of the grouper. We checked in the 90 days and 120 and 180 days. Here you can see the difference between 90 and 120. The ovarian cavity is very clear in 120 days. And the developing oogonias seems very clear here, but not in the 90 days. But come to 180 days, it's almost completely developed oogonia. So we confirmed that the gonadal sex differentiation occurred around 120 days. So the short summary, the reduced amino acid sequence of grouper neurosteroidogenic enzyme genes showed higher identities with other TDS species. Compared with the human sequence, the identity of our clone sequence was more than 63%. So the presence of developed oogonia with ovarian cavity were clearly seen at 120 days compared to 90 days. So we also did the tissue distribution of the important genes 3-beta-HSD and CYP19. So we detect the intensity of the band of 3-beta-HSD in the midbrain compared to forebrain and hypothalamus. We also detect the signal in the head kidney and the gonad. When come to the brain aromatase, we detect high signal in the forebrain only compared to midbrain and hypothalamus. We also detect a little signal in the spleen. So the mRNA expressions of the steroidogenic enzyme genes, we compare in 90 and 110, 180 and 150 days. So first gene, the CYP11A1, it's significantly increased at 110 days. And 3-beta-HSD, it's increased in the midbrain around 110 days. Also we detect in the hindbrain. In the CYP17A1, it's increased in the midbrain, hypothalamus, also in the hindbrain. So all the three genes, it's significantly increased around the gonadal sex differentiation period. And CYP19A1B transcripts, it's significantly increased in the forebrain. And in hypothalamus, it's significantly upregulated from 90 to 180 days, 150 days. But in the midbrain and hindbrain, there is no changes. And brain aromatase activity. The brain aromatase activity also similarly expressed in 110 days. Also the forebrain, here also the forebrain. But in the midbrain, no significant change was detected. In the hypothalamus, the expression was significantly upregulated. So we did the in situ hybridization, a signal for brain aromatase. We found the hybridization signal on the telencephalonic ventricle and the radial glial cells. Here you can see this is the sense and this is the antisense. This is the magnified picture of antisense. This is clearly you can see the hybridization signal was found on the radial glial cells of the telencephalonic ventricle. And also we detect the signal in the preoptic area and diencephalonic ventricle. And this is the 3-beta-HSD hybridization signal. We detect